This is RTP. This is RTP180.
Hi, I'm Rajani from the Terrio Lab, and our lab is part of the Department of Population Health and Pathobiology at NC State College of Veterinary Medicine. I've also had the privilege to work and study at NC NC State, Auburn University after graduating with a degree in veterinary medicine from India. And at the Terrio Lab, we work on a major public health concern, that's Clostridioides difficile infection. It's more commonly called as C. diff infection or CDI. And we use a combination of molecular and microbiological tools, animal models, and high-throughput sequencing to gain a fundamental understanding about this disease. And the ultimate goal is to design targeted therapeutics to treat C. diff infection in humans. And a little background about C. difficile. So C. difficile is a rod-shaped bacteria that grows in the absence of oxygen. In the light of Halloween, I'm going to point out some spooky features about C. difficile. So the feature number one is under unfavorable condition, they form spores. And these spores are highly resistant to various environmental insults, including commonly used disinfectants. And under favorable conditions, these spores germinate and form vegetative cells. And when we grow these cells in the lab on a Petri dish, they appear as medium-sized off-white colonies that have a very distinct odor. And under the microscope, they appear as rod-shaped cells. And this cell is even starting to form a spore here, which will eventually be released out of the cell. And when these cells, they multiply and they grow into high density, that's spooky feature number two, it starts producing toxins. And most of the C. diff strains, they have 1 to 4 toxin genes that help them to make the toxin. And this toxin is responsible to damage your intestine and cause disease. And moving on to, so when you ingest the spores, these spores come in contact with the bile in your intestine, and primary bile salt, which is a major component of the bile, helps the spores to germinate and form the vegetative cells. When your gut is healthy and you have all the good bacteria, which is an intact microbiome, that helps to inhibit the different stages of the C. diff life cycle that I just mentioned. So there is no toxin and no disease. However, when your gut microbiota is compromised, especially with the intake of antibiotics, so the good bacteria in your gut is wiped out, C. diff is not inhibited. It completes its life cycle, multiplies to high density, starts making toxin, and it results in disease. And this is a colonoscopic image of a healthy colon versus a diseased colon, showing all these lesions in here. And the impact of C. diff infection in humans is huge. According to the CDC, it is an urgent threat to public health, and it causes over 500,000 infections, 29,000 deaths, and over $4 billion in medical expense every year. And historically, C. diff is a hospital-acquired infection, and the number one risk factor for CDI is antibiotic intake. But over the past decade, there has been a change in the epidemiology of C. diff infection, and it's now increasingly seen outside the hospital settings within the community, and this is called a community-acquired C. diff infection. And the risk factor for community-acquired C. diff infection is largely not known. And we know animals also carry C. diff. And so are these animals actually providing an adaptive landscape for human C. diff infection? In order to answer that question, we looked at the prevalence of C. diff infection or C. diff in animals. So in total, 201 fecal samples from various animal species were tested for C. difficile, reminding you about the spooky factor number one, that's spores. So we enriched for spores in the feces, And we found that 20.8% of the samples were positive for C. diff, and of which 33 were from dogs, 7 from horses, and 2 from cats. All right. So we also looked for the genetic diversity and antimicrobial resistance, and we've to spooky factor number 2, that's toxins. So we found 69% of the isolates were positive for toxin genes, and 18 different ribotypes were observed. And interestingly, the major ribotype that we found in our data coincided with the ribotype that is the leading cause of C. diff infection in humans. And the isolates were also resistant to antibiotics that are commonly used in clinical settings, but thankfully they were not resistant to the antibiotics that are used for treating C. diff infection. And finally, we would like to use high-resolution microbial genomics in a One Health framework, meaning looking at animals, humans, and environment at the same time. So we bring all pieces of the puzzle together, so which will help us to better understand this infection and also treat this infection in both animals and humans. And that ends my presentation. I'll be happy to take questions.
All right.